Hello everyone, my name is Edith and in this video I will present part of the research work of my PhD within the Panorama project. The topic is Influence of pH on the toxicity of rare earth elements, lutecio, samarium, yttrium, and ytterbium to model organisms. Rare earth elements occur naturally in the environment and they are currently in high demand. They have become key in the production of high-tech products. Therefore, anthropogenic activities related to their extraction and management processes have caused them to increase their environmental concentrations. Because of this increased occurrence, rare elements have been recognized as critical emerging contaminants. At the moment, we still cannot fully understand their behavior and the risk they represent to the environment. The toxicity of these elements is highly influenced by many environmental factors. In some cases, they can present some hormetic effect, which means that at low concentrations they will stimulate growth. For this, they are also used in agriculture and for stock organisms. However, at higher concentrations, they can also be toxic. In this study, we focus on the influence of the pH on the toxicity of 4 elements: yttrium, ytterbium, lutetium, and samarium. We wanted to simulate 2 different scenarios, so we tested the toxicity in acidic conditions at pH 5 and at basic conditions at pH 7. We tested 3 different model organisms and exposed them to a range of concentrations to obtain a dose-response curve. Our first organism was the bacteria Alibrio fishery. As endpoint, we measured the luminescence inhibition after 30 minutes of exposure. Then, for the green algae, Rhaphidocelis subcapitata, we measured the growth inhibition after 72 hours of exposure. For the crustacean, Daphnia magna, we measured the mortality after 48 hours of exposure. In these graphs, you can see the results obtained for the exposure of Alibrio fishery. In the y-axis, you can see the luminescence inhibition, and in the x-axis, the concentrations tested. The green lines represent the pH 7 and the orange lines the pH 5. For lutecio, samarium, and yttrium exposure, you can also see a dotted line located at 0% inhibition, which means any point below indicated stimulatory effects. We can observe stimulation for lutetium at the lowest concentration at both pH, for yttrium at a middle concentration for pH 7. Samarium shows stimulation in all concentrations on both pH, and in contrast, ytterbium showed an inhibitory effect at all concentrations on both pH. Moreover, in this table, you can see the estimated 10% effect concentration, or EC10. For samarium, it could not be calculated because the stimulatory effects. For lutetium and yttrium, we obtained lower values for the lower pH, and for ytterbium, the lower value for pH 7. For Rhaphidocelis subcapitata, we have the same display of the results, only that the endpoint corresponds to an inhibition of growth. For lutetium, only the second lowest concentration at pH 7 caused a bit of stimulation, but all the other concentrations caused inhibition. For yttrium, we can see that the lowest concentrations there was very small stimulation at pH 5, but ultimately both pH caused a similar increasing inhibition, reaching 100% at the highest concentrations. For samarium, we can see that at pH 7, lower concentrations cause stimulation, at higher concentrations inhibition. However, this inhibition was maintained relatively constant. Similarly, at pH 5, we see that there is almost no effect and at higher concentrations caused more stimulation. Ytterbium had also different behaviors at different pH, as at low concentrations, pH 5 caused stimulation and pH 7 inhibition. Ultimately, at the 2 highest concentrations, both caused inhibition. For the estimated EC10s, we obtained overall lower values for the higher pH in all elements except for ytterbium. Lastly, for Daphnia magna, we can see more clearly a differentiation in the toxicity between both pH. At pH 5, the elements cause higher mortality with increasing concentrations, resulting in 100% mortality at the highest concentration. In contrast, at pH 7, the mortality was not in any case above 20%, and interestingly, mortality was 0% in all causes for lutetium. For the estimated EC10s, we can see a similar trend to that obtained for algae. In this case, only for yttrium and ytterbium it was possible to get an estimate for pH 7, and the values are lower than those obtained for lower pH, meaning that they cause higher toxicity at lower concentrations. However, the trend indicates that higher toxicity effects were caused by lower pH, while the higher pH remained with relatively low toxicity. In conclusion, from these results obtained, we could observe that pH 5 caused higher toxicity in most of the cases. This is relevant if we take into consideration some scenarios where acidic conditions can occur. For example, rainwater has a pH around 5, making it slightly acidic. Acid mine drainage can have a pH from 2 to 6, and it is also considered an entry pathway of rare earth elements into the environment. And lastly, the ongoing ocean acidification caused by the use of fossil fuels, increasing the stress on aquatic species. As for the stimulation effects, some studies on primary producers report the increase of antioxidants or protein production that helps increase their stress tolerance, which leads to beneficial effects. However, as the concentrations of the stressors increase, it starts causing damage to the cells. Therefore, the beneficial effects should be taken carefully, as the environmental conditions can influence and lead to negative and unwanted effects. Moreover, the general principle of rare earth elements exposure and toxicity is that toxicity is highly influenced by the bioavailability, which is further influenced by the complexation. By having a low pH, the toxicity might increase because the elements are more available for the organisms to interact with due to their presence as free trivalent ions. This principle is of course more complex in a realistic environment, as many other factors will affect each of these circles presented, such as the presence of organic matter, the temperature, the condition of the organism exposed, and its ability to cope with the stressor. Therefore, further studies will be focused on more complex scenarios with multiple endpoints and longer exposure time. I thank you for your attention. I hope you enjoyed this presentation, and please don't hesitate to contact me if you have any question regarding this research. Have a nice day. Bye.